Hello, everyone. I think we're ready to start. So just at the beginning, I'd like to just say on behalf of the Biochemical Society in Portland Press, I'd like to welcome you to today's webinar. This is part of the Biochemistry Focus webinar series. This has a number of different topics in different research areas in the molecular biosciences, as well as some sessions that support career development. And it's important to say that we have opportunities for asking questions at all of these webinars. So first of all, I guess I should introduce myself. So I'm Wendy Harwood. I'm head of the Crop Transformation Group at the John Innes Centre in Norwich in the UK. I've been working for many, many years, initially on GM crops and then more recently using genome editing. And to be honest, this is one of the most exciting new technologies that I think I've ever seen in my quite long career in the area. So I'm excited about this webinar. So our title of the webinar today is Genome Editing, Potential Contribution to Sustainable Agriculture. It's related to a themed issue of Emerging Topics in the Life Sciences Journal, which was actually published back in 2017. And of course, things are moving so quickly in this area. A lot has changed since that time. So genome editing, particularly the CRISPR-based technologies, continue to generate huge excitement in the opportunities that they offer for improving agriculture. And we're going to hear some examples today. So, I mean, the first genome edited crops actually have already been made available commercially. But, today we're going to explore this technology a little bit more in the contribution that it could make to sustainable agriculture. Okay, so we have two speakers, which I'm really looking forward to hearing from. And they are experts in using genome technology in either crops or livestock, respectively. So our first speaker, Dr. Joyce Van Eck from Boyce Thompson Institute, Ithaca, New York. And our second speaker, Professor Bruce Whitelaw from the Roslin Institute in Edinburgh. So what we're going to do is have both presentations, and then there will be an opportunity for you to pose questions to either of the speakers. So I think you can see the details on the screen. There is a question box that you can put your questions in. So please do, if you think of questions during the talk, please add them in. You don't have to wait until the end, and then we will try to get through as many questions as we can at the end. So each of our speakers will speak for a maximum of 20 minutes. Okay, so without further ado, let me introduce our first speaker. So Dr. Joyce Van Eck is Associate Professor at the Boyce Thompson Institute. This is located on the Cornell University campus in Ithaca, New York. And she is a leader in the development of using biotechnological approaches to study gene function as it relates to crop improvement. And she's particularly interested in using genome editing technologies to fast track improvements in crops, specializing in the more underutilized crop species. And you will hear examples from her. Okay, so I think without further ado, I will hand over to Joyce. So Joyce, if you can share your presentation, we'll just check that everything is okay. That's perfect. You're all set to go, Joyce. Looking forward to it.
Okay, thank you. All right, well, I'd like to thank Wendy for inviting me to tell you about our work with this little yellow fruit, which is goldenberry, and we're also working with a fruit called ground cherry. So these are some points that I'll cover during my presentation. I'll tell you about how we got started working with the physalis. I'll then move on and talk about the characteristics that need improvement. And these are being driven by our National Science Foundation, or NSF funded project, and also by feedback from our community scientists. I'll tell you about how we're fast tracking the improvement with gene editing, specifically CRISPR-Cas. And I'll end by telling you a little bit about a community science project that we have where we're working with farmers and home gardeners to obtain their feedback firsthand on the characteristics that they feel need improvement, and also to heighten the visibility of ground cherry and goldenberry. So how do we get started working with the physalis? Well, my collaborators, Zach Lipman and Mike Schatz, and I had a project a number of years ago with tomato, and we were looking at the genes and networks involved in plant growth habits, fruit characteristics and also inflorescence architecture, so flower structure. We were using a reverse genetics approach to knock out gene expression and then looking at the resultant phenotypes. And we were using CRISPR-Cas to do this. And through this work, we came to realize that CRISPR-Cas could be a powerful tool for fast-tracking improvement, in a sense, fast-tracking domestication of underutilized species. So in our next round of funding, we decided to put that idea into practice. And we decided to stay within the Solanaceae family. And as you'll see here, this is a phylogenetic tree of some of the Solanaceae. And we wanted to work with a species that had very little, if any, crop improvement. And we decided to work with the physalis. And the physalis that you might be most familiar with is tomatillo. But we chose to work with ground cherry and goldenberry. And the reason for that is that we felt that in addition to being able to answer our basic fundamental research questions, there were also potential for these fruit to become specialty fruit crops in the US, but they needed improvement to be adopted on a larger agricultural scale. Now, ground cherries in the US, we find them in farmers markets, and goldenberries, we find them in grocery stores, and in other countries, you're most likely to see goldenberries. Now, people often ask me what they taste like, and they expect them to taste like cherry tomatoes because they look like cherry tomatoes, but they're actually sweet. So ground cherry has a simple sweet flavor, whereas goldenberry has a more complex sweet tart flavor. And we also have some accessions or plant types that have slight flavors of coconut, mango, and apricot. So some characteristics of interest is that to us, they're underutilized crops in the US. They're more popular in other countries. And as a matter of fact, Colombia is the major exporter of goldenberries, second to their export of bananas. And you'll see down here in the right hand corner there are some goldenberries that I took a picture of in our local grocery store, and they're in with the raspberries and strawberries and other types of berries. They have a good nutritional content, vitamin C, beta carotene. They have anti-inflammatory and antioxidant properties. And these properties have lent themselves well for these fruit and plants in general, and the physalis, to be used for medicinal properties because of these compounds. And also they have compounds for plant protection with their withanolides and phthalans, which have steroidal properties. And these properties, these withanolides in particular, have medicinal properties as well. As for characteristics that need improvement, plant growth habit is the #1 obstacle for ground cherry and goldenberry to become adopted on a larger agricultural scale. Now, this is an image of our field and this is goldenberry. These metal stakes that you'll see are 6 feet tall from ground to the top of the stake. And this is in July. So you can see how large these plants are just in July. And we end up having to trellis them. And we use this twine, and it's called the Florida weave style of trellising. But you can imagine on a larger agricultural scale, this is a lot of resources and time that farmers would have to put into this. As for ground cherry, you'll see the ground cherry in the foreground, the goldenberries are in the background. Now, ground cherries don't grow as tall as goldenberry, but they do have this very sprawling growth habit. And it's really difficult for farmers to grow those as well if they didn't stake them. The other problem with ground cherry, and the reason it's called ground cherry, is that it drops its fruit to the ground. And you can see, this is one of our greenhouses where we've had some planting, and just see all of these fruit on the bench and on the floor. They drop at different stages of ripening. And again, you can imagine on a larger agricultural scale, this would be difficult for farmers. And actually, we've had some farmers tell us that unless we could correct this fruit drop problem, they won't grow them. But their customers love the flavor, and they would grow, the farmers would grow acres of them if they didn't have this problem of fruit drop. Now, the reason for that fruit drop is this little region here on the pedestal. It's like an elbow, and this is the abscission zone, and this is where it causes the fruit to drop. So we are currently studying the biology of this region to determine how we can correct that problem of fruit abscission. The other feedback that we've gotten from our community scientists is that the ground cherry fruit is kind of small. So they would prefer to see it more the size of goldenberry. So that's another project that we're looking at, is ways to increase the size of ground cherry fruit. Now you can see here we have ground cherry, we have some from the Netherlands and from Poland, and you can see that size difference compared to golden berries. This one is an accession from South Africa, and this one here is an accession from India. Now I'm going to move on and tell you how we're using gene editing to fast track that improvement. But before I do that, I have to tell you about our plant regeneration and plant transformation systems. Now, in order to do gene editing, we need a method to be able to introduce those gene editing components into plant cells. And the first place we start at is in something called plant regeneration. And we started with ground cherry because ground cherry is a diploid, whereas golden berry is a tetraploid. And we felt that doing our first editing in a diploid would be much easier than trying to do gene editing in a tetraploid, where we'd have more copies of the gene to edit. So for ground cherry, we looked in the literature first to see if there was anything reported for plant regeneration, and there wasn't. And then we looked to see if there were any closely related species, and we found that there were methods for tomatillo and another physalis, but those methods didn't work for ground cherry. So since ground cherry is in the same family as tomato, and we do a lot of work with tomato, we decided to simply try our tomato method. So we start with germinating seeds on a medium, the culture medium that contains a gelling agent to solidify it, sugar, sucrose, and also salt to support plant growth. These are the germinated seedlings, and for tomato, what works the best are the cotyledons for plant regeneration. But what we found for ground cherry, what worked, the cotyledons didn't work, but what worked really well for plant regeneration were these hypocotyl sections. So we cut those hypocotyls into small sections, and we placed them on a medium that had similar components to what we use for the germination. But in addition, we added a plant growth regulator or hormone that induces cell division and then gives the cells the signal to produce shoots. So you'll see here in this image, we have a good number of shoots that are forming. We cut those shoots are then moved to a rooting medium. They root readily. We don't need to include any sort of plant growth regulator or hormone. We then take cuttings, we root them into test tubes so we have individual plants. And then it's this point that we'll do our analysis. But again, this is just our plant regeneration before we even start looking at doing a transformation and introducing the gene editing components. Once we have those rooted plants, we have to transfer them to the real world, transfer them into soil. And if we didn't cover these plants with either a plastic bag, and in this case, this is a plastic water bottle, they would wilt and die. And the reason for that is you saw those plants in the test tubes, and it's a very humid environment, and the stomates are wide open, and the cuticle hasn't developed. And if we didn't cover them, they would wilt and die. So what we do is we take these water bottles that have the bottoms cut off, and we cover those plants when we take them out of culture. And when we take them out of culture, we also have to wash off that medium because there's sucrose in there, and a lot of microorganisms would grow. After a few days, we remove the cap to give them a gradual acclimation. And then a few days after that, we remove the bottle. And you can see how well that worked. This is the same plant several weeks later, and we have 100% survival rate with this transfer to soil, which is an important part for our project to make sure we can start evaluating the phenotype. Once we had that plant regeneration system worked out, we then moved on to transformation, which is the method to be able to introduce our vectors or gene editing components into plant cells. And this is work done by Carrie Swartwood. So we have those hypocotyl sections that I showed you in that last slide. We infect those with agrobacterium, and that's an agrobacterium that transfers our vector and gene components into the plant cells. We're using a strain called AGL1. We transfer those to a plant regeneration medium that has the plant growth regulator zeatin. This medium also contains kanamycin, and I'll explain why that is when I talk about the vector in the next couple of slides. And then we transfer them to rooting medium, as I showed you, and then onto soil. So this is an example of a vector or a construct that we use for working out a transformation system, because before we jump into doing gene editing, we need to make sure that our introduction into the plant cells is working. So this is a vector. The agrobacterium recognizes these left and right borders to cut out of the plasmid, and this is the part that gets introduced into the plant cells. So I mentioned that in kanamycin in the medium, and the reason we use that is because every cell in that hypocotyl has the potential to go on and regenerate plants, but not every cell gets infected with the agrobacterium. So we want to have only the plants that have received our vectors to go on, our cells to be able to go on and divide and make plants. This way we really don't have a lot of plants that we would then have to evaluate. So to do that, as part of our vector system, we have this NPT2 gene that confers resistance to the antibiotic kanamycin, so that only the cells that receive this vector should go on and divide and produce plants. So it's really a way to keep down the number of plants that aren't transformed. And a part of this is we use something called a green fluorescent protein. It's GFP, and that helps us track the progress of the plant transformation. We do electroporation into our agrobacterium. We grow that overnight, and then we do the transformation experiment with those hypocotyl segments. Now, these are some tools that we use for that agrobacterium. So this is these are agrobacterium colonies that were a result after electroporation of introducing that vector system into the agrobacterium. We then take a colony and we put that into this bacterial medium. It's incubated overnight on a shaking incubator, and then the next day we have the agrobacterium has grown. We measure the optical density, and then we do the infection of the hypocotyl segments. So I mentioned that we're using GFP, and that helps us track progress. You can see in this image here, this is a hypocotyl segment that's been on that plant regeneration medium for about 2 weeks. These cells have divided at the end of the hypocotyl, and they're making something called callus. And you can see that we have GFP expression. If we didn't see GFP expression at this point, we would go back and tweak the infection process, because there's no sense in going through the whole system if we haven't had good delivery into those plant cells and expression. Over a bit more time, you can see there's some callus here. That's a green fluorescence. From that callus, we regenerate shoots. This image here shows a white light type of imaging, and then here on the right is a fluorescence imaging, and you can see the green fluorescence expression in those little shoots. We grew the shoots to maturity. We put them in the greenhouse. When they produced fruit, we decided to see if the expression would continue in the fruit. And you can see here is a ground cherry fruit, which is one of my favorite images of my career, expressing GFP. We saw GFP in the husk, and we also extracted some seeds, and we could see the GFP in the embryos, and that was an indication to us that our transformation method worked all the way through to transmission to the next generation. So it's at this stage we thought, all right, we've got a good plant regeneration system, a good transformation system. We can now move on to our gene editing. This is a vector similar to what I showed you before. It's got the NPT2 gene. We have the Cas9 that does it's the endonuclease. And our guide RNAs are driven by the Arabidopsis U6 polymerase promoter. You really only need one guide RNA to see gene editing, but since we were doing a reverse genetics approach and we really wanted to knock out expression, we used 2 guide RNAs spaced about 50 to 100 base pairs apart. We introduce that into the agrobacterium, and then we do the transformation experiment, as I discussed before. So once we had that method all worked out, we were then able to move into targeting the traits of interest. Now, in the interest of time, I'm only going to tell you about a few of the genes, and I'm going to talk about plant growth habit and fruit size. So for plant growth habit, one of the first genes we targeted was something called self-pruning. And I need to tell you a little bit about something called sympodial plant growth. So in tomato, if you've grown tomatoes, you know there are some plants that grow wild and continue to grow and they're very tall. And there are other plants that are more compact, and they're called determinate. Well, the reason for the indeterminate type of growth, that wild continued type of growth, is something called sympodial plant growth. And what happens is you have this reiteration of 3 leaves and fruit, 3 leaves and fruit, and that continues to go on and progress in that manner. And that's because of this gene called self-pruning. And self-pruning is an anti-flowering gene. But back in the late 20s, a grower identified a more compact plant in his field. And over time, they identified that this was a mutation in this self-pruning. So this mutation in self-pruning caused a more compact growth habit or determinate growth habit. Some of the hallmarks of SP mutants, you get bursts of early flowering and there's shortened sympodial units. And then breeders took this and started to breeding it into tomato commercial lines, and it really revolutionized commercial tomato production by making those plants more manageable to grow. So we decided to recreate that SP mutant in ground cherry. We found the self-pruning counterpart in ground cherry. In this upper image here, you can see the top line is our wild type sequence, and then these are some of the editing results that we saw. We have a deletion here and some insertions. And the image below shows you the phenotype of some of our CRISPR lines. The image here on the left is a wild type. The stakes are about 4 feet tall. And you can see that the self-pruning, creating that mutant, definitely caused a really amazing, more compact plant habit in ground cherry. Now, what we do with our edited lines is we then, after we've evaluated them in the greenhouse, they go to the field and I collect seed, and then those go into the field, and we evaluate those for the persistence of the phenotype and also possible any negative effects on other characteristics like yield or fruit size. So we're at that point right now. As for fruit size, there are a number of genes we can target and we have targeted, but I'm going to tell you about this one called clavata1, since that was the first gene we targeted. Now, clavata1 works in concert with another gene called WUSCHEL to keep everything normal in the plant, keep that shoot meristem maintenance very normal. You can imagine that a loss of function of one of those genes would cause an increase in the size of the shoot, the floral meristem, and floral organ number. So when we knocked out clavata1, that's indeed what we saw. The first sign was that we have an increase in the number of petals. For the wild type here, you'll see there are 5 petals, whereas in this one clavata1 mutant, we have 7 petals. What was exciting to us is we actually saw an increase in fruit size. These are the wild type here, and these are some of the CRISPR fruit. And we actually had a 24% increase in fruit weight. And one of the differences we saw that in this fruit that, in the wild type, there's something called bilocular, where there's the cavities that hold the seeds, and they had 2 locules, whereas in the CRISPR mutants, we had 3 locules. But overall, an increase in fruit weight, which again was very exciting to us to see that we could affect that trait. An experiment that it's very early stage is the fruit drop. As I mentioned before, it's a real problem in ground cherry. And this is work that was done by a postdoc in our group, Nathan Ream, who's now has a position at a company here in the US. And the work is being picked up by a new graduate student in our group, Elise Tomaszewski. So what Nathan did was he started out with doing RNA-seq of that joint region that I showed you earlier. And we had a stem control in addition to the early joint development, mid and a late joint development. We got all that RNA-seq data. This just shows one, for those of you who've done RNA-seq, it generates a lot of data. This shows just one part of the data. Nathan then went ahead and did what's called a heat map of that data. And that the way this works is that each line represents a gene that's being expressed. Yellow indicates a high level of expression, and blue indicates a low level of expression. And you can see the differences between the control, the early, middle, and late stages. Now, Elise is picking this up and doing more evaluation of the RNA-seq data to help us identify genes to be able to target with CRISPR and then look at the effect on the fruit drop. So I want to take just a couple minutes just to tell you a little bit about our Physalis Improvement Project, where we're working with farmers and home gardeners and consumers to get their firsthand feedback on what needs to be improved. And we're also using this as a way to do outreach and science communication about the Physalis. So we have a website, and if you're interested, just search for a Physalis Improvement Project. I have a Twitter account, and we have a Physalis Project. And we provide seeds of the unimproved materials, so the wild types of ground cherry and goldenberry of different accessions. We provide these to people, they grow them up, and they give us feedback on how they're doing, and we've received very valuable information. Last year, we had 750 participants in 43 different states, and you can imagine just getting that information across regions, how useful that is for our project. So in summary, I've shown you that CRISPR-Cas is a very powerful tool. We did that work with the SP and the clavata. It took 2 years, and that included working out the transformation system. We're moving on to goldenberry right now, and we have some new directions. We're looking at metabolites and volatiles, specifically working with the withanalyzed. And with that, I'd like to thank the members of my group, Savannah, Nathan, Carrie, and Elise, and our greenhouse group, who do a great job growing our plants. My collaborators, Zach Lippman and Mike Schatz, our community scientists, and our funding agencies, because without them, we wouldn't be able to do this work. And I'd like to thank you for your attention.
Okay, hope you can hear me again. Thank you very much, Joyce, for that. That was brilliant. And it's so nice to hear examples in some of the more unusual crops and the ones that we focus on most of the time here in the UK. So yeah, please, if you have any questions for Joyce, do pop them into the questions box and we will come to them after our second talk. So thanks, Joyce, and look forward to chatting to you a bit more after the second talk. So now we'd like to move on to our second speaker. We're moving from plants to animals. So our second speaker is Professor Bruce Whitelaw. So Bruce holds the Chair of Animal Biotechnology at the University of Edinburgh. He's also the Interim Director of the Roslin Institute, which is a BBSRC strategically funded institute. That's embedded into the Royal School of Veterinary Studies at the Easterbush campus of the university. Bruce has a lot of responsibilities. He's Dean of Research and Innovation, Chairman of the Roslin Innovation Centre and Director of Roslin Technologies Limited. It's a very distinguished scientific career, many papers, patents, but he really focuses on the application of genetic technologies in farmed animals, and currently one of his main areas of focus is genome editing in farm animals, and he's going to give us some examples today. So Bruce, we look forward to your talk. If you'd like to share your screen, and I'll just let you know, yeah, I have that there. That's all looking good. So Bruce, I'll hand over to you.
Okay. So thank you, Wendy. And thank you everyone for tuning in here. As Wendy said, I have many roles to play, but one of the most exciting and pleasurable one is when talking about the science that we do. Today I'm going to tell you about some of our genome editing work in livestock, but first I will give you the sort of context of where that is. I'm then going to give you details of one specific project to do with disease resistance, and then broaden it slightly at the end to indicate where we're going with this. So livestock agriculture faces a number of challenges all coming at the same time. It is an important part of how we secure food supply for societies around the world. One of the key challenges we have is to reduce the burden of disease, infectious disease in particular, and we have to do that while delivering towards a carbon zero agenda, looking at the challenges of climate change and increasing the animal welfare of animals on the farm. So to put that sort of succinctly, more efficient livestock production from less resources with enhanced welfare. So a lot of challenges there. And I believe, and I hope to explain over the next few minutes, that genetic engineering, in particular genome editing, but also the more traditional GMO transgenic technologies offer some options there. So the two examples I'm going to give, and I'll describe one in detail and one just in passing at the end, relates to pigs and to cattle. The pig project is looking at disease resistance, and I believe this addresses four of the challenges I indicated a few minutes ago to you: food supply, reducing disease burden, obviously addressing carbon zero in that we can generate more healthy animals, and also looking at animal welfare. The second example I'll just mention at the end is to do with climate change or heat tolerance. And again, I believe this addresses many of the challenges facing livestock agriculture. The example I want to give with pigs is perhaps the lead project coming out of the Roslin at the minute. and it's to do with a disease called porcine reproductive and respiratory syndrome. This disease has a huge impact on pigs, animals which are pregnant, associated with abortions, increase in stillborns or weakness, and in some cases infertility. With young animals, and even older animals, there are a number of respiratory syndromes associated with this, which can be life-threatening, and I'll come up with some numbers in the next slide. The virus, this disease is caused by a virus. The virus is called porcine reproductive syndrome virus, and there are vaccines, but they are at best partially effective. And the last thing, which I think is important, is this is a global problem. It's not a virus which adheres to any geographical boundaries. There are three costs, I suppose, to this disease. One, we can capture numerically or value, depending on which side of the pond you're on. It's either approaching $1 billion a year in the US, or well over €1 billion if you're in Europe. As indicated, there are a variety of symptoms associated with infection, but the baseline for this is that the virus destroys a large proportion of the macrophages as part of the immune system of pigs. In an area where the virus is prevalent, you can get up to well over half of the herds carrying the virus, and this has a devastating mortality rate in those herds. During the acute phase of an adult animal, if you want to call it that, we can see anything up to 15% mortality. However, this goes up to 80% when looking at young animals. Not only is this an impact on the animals themselves, but there's also an impact on the farmers that are rearing those animals, and in many cases leading to financial ruin and heartbreak as the virus comes through their animals, their populations. We know a reasonable amount about this virus. We know in particular from in vitro cell culture studies, looking at cells which are permissive or non-permissive for virus entry, we know that there are two proteins, CD163 and CD169, which are involved in entry of the virus. And I'm going to focus today telling you about our progress with CD163. We also know the structure of this protein, and it is a transmembrane protein with these string of beads or modular domains sitting outside the cell. And we know more than that. We know that each of those domains has different functionality attached to them. In particular, we know that domain 5 is involved in PRRS infection. So we focused our attention on domain 5. And this little cartoon here hopefully will show that to you. The virus, the protein intact CD163 on the cell surface, and the virus will come along and bind to it. We used 2 guide RNAs to remove exon 7, which is the exon encoding for domain 5, leading to a truncated protein. And the hypothesis was that the virus now would not bind to the cells that carry that truncated protein. This is a busy slide, and you don't need to identify the detail on this. The 3 pigs show 3 different phenotypes, non-edited, heterozygous, one allele edited, or the homozygous, both allele edited. The basic take-home message is here, apart from a reduction in the size of the protein as shown in the Western blot on the left, the amount shown in the histograms between these 3 genotypes is the same, and on the graphs on the right, the ability to traffic to the cell surface, the ability to differentiate upon induction into a macrophage in vitro, and the ability to scavenge heptoglobin and hemoglobin, which is the biochemical function of CD163, are the same regardless of which genotype you are looking at. More importantly, when challenged by live virus, these animals are resistant. So the orange graph, part of the graph here, shows your non-edited animals, whether they're heterozygote or non-edited, not edited at all, while the edited animals are the green lines along the bottom. And in our small-scale pilot study, we showed that these animals are entirely resistant to infection by the virus. This work started 10 years ago, and the first phase of it was performed by the Roslin Institute. We have done this project in partnership with a pig breeding company called Genus PIC, who have now taken the project on with licenses from the university to do so. And Genus are now looking at a 4-year timeline, 3-4-year timeline, to now progress the project in partnership with the FDA to a product that we could see in our fields and farms. This shows you, this project I think shows you the elegance of genome editing, a simple removal of a few hundred bases, including an exon, leading to resistance from infection from a virus. We are looking at other pig viruses, we're looking at poultry viruses, and we're looking across ruminants as well. I suppose the one that catches the attention on this page would be influenza, both swine and avian, and colleagues at the Roslin in association with Imperial College London are making progress there. We're also looking at other poultry diseases such as Marex or Newcastle and FMDV in ruminants. We believe that we can identify candidates which will allow relatively simple genome editing approaches to confer or alter the susceptibility of those animals to virus infection. But the same idea, the idea that you can incorporate a small deletion or change of sequence or using genome editing tools, we can apply it to other traits. And one of the traits we're looking at is to do with heat tolerance. The trait's called SLIC. It involves the prolactin receptor. Mutations been found in a number of cattle breeds, one in particular, the Senepol. And American company called Excelligen, part of the recombinetics family, have edited this change into Red Angus. And my colleague here at Roslin, Simon Lillico, has done the same into Aberdeen Angus at the Institute. And we are looking to these animals growing up and being able to assess their impact on heat tolerance. The Senepol trait is associated with reduction in the need for water through to less water waste loss through respiratory activity of those animals. So I hope in these few minutes I've given you a really good example, which is the PRRS pigs. And I believe there are many more coming. And I'm very passionate about the fact that genetic engineering, both genome editing and the more traditional GMO transgenic tools, really do offer some options for taking our science into the field and helping create the type of future farming that we want for our livestock. And at that, I'll stop, Wendy, and I look forward to any questions.
Okay, thank you so much, Bruce, for that. And it's such an exciting area. Okay, so just to remind everybody that if you've got questions for either of our speakers, please just write them in the question box. We've got a little bit of time left, and so what we'll do, Joyce, we can't actually see you at the moment. I don't know if we can get your camera. Well, oh, there you are. Perfect. We can see you. Great. I think there were some issues. We couldn't actually see you during the talk, but we had your slides, so it didn't matter. Okay, so if we can just kick off with some questions. Can I start, Joyce, with you? And just I've got a couple of questions that have come up, which are slightly sort of just technical questions. So if I can just ask those. So the first one is from Leela Grandgeorge, who says that she's obviously working in tomato and said that tomato regeneration is known for having a significant occurrence of escape shoots, so where non-transformed shoots grow from the callus despite the presence of a selective agent. So she wants to know, did you see evidence of escape shoots occurring during your ground cherry regeneration?
We don't see escapes for our tomato, a large number. Occasionally, we might have one or two that comes through, but I'd say about 98 to 100% of our lines are indeed transgenic, both for tomato and for ground cherry. A lot of our work has been geared towards, making that selection very efficient. And I don't know if for their genotype of tomato that they did this, but what we found is instead of doing what's called a kill curve before transformation to determine the most efficient level of selection, we do that in combination with transformation, because we found that what we identified as the best concentration of, say, kanamycin before transformation didn't always translate to after. So we really, from then on, for years now, have done that in combination with transformation. So might be something she should try.
Okay, well, thanks, Joyce. And just one more. This is from Aska Noreen, another question that's come in. She says, yeah, wonderful talk. Thanks, Joyce. She's curious about the editing events by Cas9, whether they are homozygous or not. And secondly, how are you able to get Cas9-free plants? Could you say something about that?
Sure. Okay, so when we do an editing experiment, we see what's called monoallelic or heterozygous and biallelic and homozygous. So we get an array of edited, and our homozygous types are actually the lowest percentage of what we see in the biallelic or the highest. And then what we do is we then transfer our plants to the greenhouse, we collect seed, and then we evaluate those seeds for the presence of Cas9, and only the material that's Cas9 negative but has the edits is then moved forward to the next generation. And we do that simply by a PCR analysis, and anything that's PCR positive for Cas9 is not moved forward. Okay, no, thanks for that, Joyce. Okay, please keep putting your questions in, and I'll keep an eye on the questions box that I've got here so I can see them all. So please keep doing that. You can see the box just to remind you. Bruce, can I just, I was thinking as you were talking about the disease-resistant pigs, the PRRS example, and you did show some data that there were, there appeared to be no effects other than the virus resistance. But was there any? Did you notice anything at all in these pigs? Was there even any other characteristics? Was there anything at all that varied in your edited pigs compared to your controls?
Good question, Wendy. The answer is no. The caveat is we did a small scale proof of concept study, so not many animals. We've had those animals through to the third generation. We don't see any obvious impairment in reproduction or any other trait, visual trait attached with it. The concept has taken, it's moved from the academic arena and moved into the commercial sector, and genus PIC are currently producing large number of animals to allow a more comprehensive cross survey of traits. To date, they have not seen any deleterious or unwanted aspects to the edit. There is another caveat to it, in that although we know the gene and the protein, and we've got information about the protein, we only know some. I presume we only know some of the biochemical activities of that gene, and there may be aspects which we haven't looked at yet. But certainly from our proof of concept pool, visually they are as piggy as a pig can be, and the feedback I'm getting from the studies performed by genus PIC is that the animals are indeed staying with that and are looking very, very promising.
Okay, now that's great to hear. So I see Aska Noreen also has a question for you, Bruce. I'm not quite sure about this one, but this probably make more sense to you. So the question is regarding the recombination factor in viruses. is that enough for you to go on? That's all I've got. I don't know whether the
there's not much to that question. I'm assuming the question relates to, will the virus escape our strategy? I'll start off by saying if we use that question to stop us trying to mitigate any virus infection, we wouldn't do it. And I draw your attention to SARS-CoV-2 and the fact that we have a plethora of vaccines and quite happily sticking them into ourselves. And yeah, it's the same challenge. That's not a reason to do it. Having said that, what's important, and depends how much you know about the PRRS virus, there are different strains that exist globally, and the data to date would say that the mutation we put into CD163 is sufficient to protect against all those strains. As a bigger challenge or bigger way of questioning this, I think we feel we've hit a sort of jackpot with CD163 in that it confers resistance. And it may not be the case when we look at other genes for other viruses. We may just be able to reduce the severity of the disease or the sensitivity of the animal to infection. And I fully suspect that going forward, we'll be looking at combination of strategies to control a disease as such. And it may be that you would combine some forms of editing or trait conferred by editing with some forms of management or indeed veterinary intervention to give you the protection you want. And we have modeled that in silico, and I've got interesting data which hopefully will see the light of day very soon in the press. But the answer is, as soon as pathogens are in a battle with the host, and it's a to and fro that goes on, and that is just the way that pathogens evolve. So yes, there's a possibility of escape. We are fairly confident that because it looks at all the strains, that's not the case, or will not be the case, and we're doing studies to actually try and come up with numerical data to support that.
Okay, so thanks for that, Bruce. So I've got one question here, which I think could be to either of you, and maybe you both like to comment. So this question is from Brij Baja, and the question is, as we're constantly updating our knowledge about the effects of nutritional components of foods on the human body, how does one know or research about all the effects of GM foods? I'm guessing this could be GM or GE foods. Would anyone like to make a comment on that?
So I would say that there is quite a bit of information out there. When you think about GMOs, it's been around for 20 some years that crops have been out there, and they've been well studied, and you can find the information. I just want to say that you need to be careful about the information that you're seeing to make sure it's a well-balanced presentation of the information. And there's also different, through online and the internet, you can also find information about that. But again, I think you just need to be, be critical about what you're reading.
I'll add to that. Thank you, Joyce. So any food, whether it's a plant or an animal, will go through the current food safety regulations wherever you happen to be on our planet, and those regulations look at issues of toxicity, although I think that's really hard to imagine if it's going to be toxic for the person who eats it. It's going to be toxic for the animal in the case of animals, so I don't see that as, but allergens and so forth will come under current regulation, which keeps our food on our plate safe. With regards to whether an animal let's take our PRRS pigs, which has a very tiny bit of less DNA compared to other pigs, and indeed that degree of variation is really, really small compared to the natural degree of DNA variation that happens between animals through copy number variation and so forth, it's insignificant in a way. That's still just DNA, and we have been eating DNA, well, forever.
Yeah. Okay. I'm just keeping one eye on the questions, so I think I get time to sneak in another question here, actually. So I was just thinking, Bruce, as you were talking, you mentioned right at the end about the other viral diseases that you might be able to tackle using using a genome editing approach. And I mean, that's quite, some of those are really, devastating diseases. So I just wondered, what's your assessment of how important this technology could be in agriculture in terms of animal welfare and being able to tackle some of these diseases in the future? Where have you got any comments? Where do you see it going? How wide could this be? How wide could the effect be?
So I think it'd be a seminar in its own right. Various answers to that, or various levels of answers. I mean, personally, I think it's going to be huge. Will it tackle every trait? Will it tackle every problem that we have? No, it won't. It will be specific ones which you can have an impact with. Now, there are, I suppose, two technical jumps which would maybe broaden the scope of it. One technical jump which we really are already engaging with, and that has been able to multiplex the editing, been able to efficiently create many different edits and many different loci within the genome. And for those of you who've been following Church's work, you'll realize that that's possible. It's still challenging in animals, especially large animals, but it's possible. So that could broaden it out to traits which are governed by multiple genetic loci, and indeed combinations of genetic targets within that would broaden out. I also think that at the minute, how we work with these reagents is we introduce them into a fertilized egg, the zygote, and animals are produced from that, and the efficiency is spectacular compared to the older technologies. You can use cloning, somatic cell nuclear transfer. We don't at Roslin, but you can do that. But all of these revolve around very tried and tested delivery vehicles, either a small injection needle or some sort of chemical to allow you to transfect into a cell or introduce into a cell grown in culture. If we could see dramatically new delivery vehicles or delivery strategies, then I could see this also increasing in scope. So to summarize my answer, yes, I do. I think it's transformative. I think we will see a number of examples of that in disease resistance, but then broadening out into other traits such as heat adaptation and similar. At least for the foreseeable future, it won't be applicable to every trait.
Okay. Yeah, it's interesting you mentioned that moving to multiplex editing. I mean, certainly we can do that now in crops. We've sort of successfully made double, triple knockouts at the same time, very successfully in different crops. So yeah, I agree with that. That's one exciting way forward. Joyce, so as you were talking, you just mentioned sort of in passing growing your genome edited plants in the field, and I just wondered so in the US, what did you need to do in order to grow them in the field? What was the process that you had to go through?
So there, through our regulatory agency, through the USDA, ARS, there's a biotechnology division, and we first submit an application that details all the information about what we introduced, the outcomes, and that's all done online, and then that's evaluated. And we get feedback about whether we could put that into the field or not. And that part of that process is also having the local United States Department of Agriculture, USDA agents come out to our fields and assess the fields to make certain that we've got some isolation from other maybe related crops. And then as a follow-up, we have to the following year, the fields are disked to make sure there isn't any remaining material from the previous year. So.
Okay. Yeah, that sort of brings me on to another area that really interests me. And again, Joyce, you mentioned it at the end, which is sort of your outreach work and the importance of that. And I mean, I don't know, I mean, you could be, and it's a question for both of you really. So what do you think is the best way to actually get, information on genome editing, and, to get the details across in an accessible way to the public? Do you have any thoughts on that? You mentioned your website, Joyce. I'm just wondering if you've got other thoughts on, the best way to tackle that.
I think for me, whatever opportunity I can take to interact with the public, or popular press, I've given a number of interviews about what we're doing, and really trying to learn from what happened or did not happen when genetic engineering started, where there wasn't enough communication and transparency, or about what the technology is. And when we for our project, when we talk about the the underutilized species and improving them, we have a conversation on our site about we're starting to approaches for crop improvement include plant breeding, mutation breeding, and in a science communication way to make it accessible to people. And what was really rewarding for me is that through our program, this is 3 years now, I've actually had people get in touch with me and ask me if they could have the gene edited lines because they see the improvements. And I thought, well, we're really making progress. And I think the other thing that helps gene editing is the application to human diseases, where like sickle cell anemia, where people are seeing benefits. And but yeah, I think it's just important to get out as much as you can, whether it's through the popular press or meeting with people and talking about it.
Yeah, I noticed we're getting very close to running out of time. I've just got one at the end. It's just a technical question for Bruce that I've just picked up from the questions here. This is from Elaine Lynn. So the question is, in living organisms such as pigs, how do you ensure the edited DNA is present in every cell? Or is this only possible in germline editing in the stem cells? I don't know if Bruce, you'd just like to comment on that one.
Yeah, so there are two methods used to produce these animals. We inject into the fertilized egg, so the single cell embryo, and therefore the animals produced, or at least the vast majority of the animals produced, carry the edit in all their cells. Derived from that single embryo. If you use somatic cell nuclear transfer or cloning, you're introducing cells that have a single edit in it, and the animals derived from that single cell. So you ensure it that way. There are examples out there where founders are mosaic, and there are a variety of strategies now being developed to try and minimize that.
Okay, thank you. I don't know, so we've literally got 2 minutes left. Bruce, did you have any thoughts on the outreach issues just while we were still?
Yeah, so I think and I echo what Joyce said. I mean, there's no one specific route. We need many people to enter into this. The ones which I find particularly most rewarding are the ones that have a dialogue. One of the examples that we have at Roslin is what we call the East to Bush Scientific Outreach Centre, where we have dedicated staff who are interacting with schools across the country and other groups, and giving time to have that sort of dialogue. Yes, we should use the media. Yes, we should websites, and yes, we have sessions at public events and so forth, and they're all useful. And all voices are good, but what's essential is that we have a dialogue around this so that we can explore each other's thinkings and concerns and hopes.
Yeah, I think that's a very good point to leave it there. Okay, so all that remains for me to do is just to thank everyone who's been listening in, and hopefully all those people who will be listening later online as well. And particularly to thank our speakers, Bruce and Joyce, for doing such a fantastic job and dealing with the questions so well. It's all gone far too quickly, but thank you so much. And I'd just like to say to those listening, if you've enjoyed the session today, please do have a look at the Emerging Topics in Life Sciences website. There's a lot more related reading there. This series of webinars always welcomes suggestions for new topics. So, you know, if you have an idea, again, the information is in front of you. Please do, you know, feel free to submit a proposal for an upcoming webinar. And of course, you can see all the past webinars, and you've got the details there, either on the website or the YouTube channel. So, okay, there is, yes, the next, you can just see here the details of the next webinar that's coming up, which is very topical, COVID-related one, tackling SARS-CoV-2 biochemistry. And you can see the details on the slide in front of you if you're interested in listening into that one. Okay, so just at the end, I mean, it's, you know, it's been an interesting time. I mean, I think everyone's doing so many webinars and things at the moment. It's been more difficult for us to do things face to face and in person, but it's been really, really important to keep in touch. So if you would like to, you know, stay connected and engaged, then consider, you know, the Biochemical Society is one of the options for that connection and engagement. Okay, and there's a little bit more information on the slide in front of you. So I just need to say thanks again to everybody, particularly big thanks to the organisers behind the scenes who've made it all work. Thank you, and big thanks to Bruce and to Joyce for joining us today. So thank you very much. Okay, goodbye everybody. Bye.